So guys, why don't you go ahead and get out your books and our story, the colonization story. That is what we will go over. I wrote the exam last night, and so I have it fresh in my mind what we're doing.
Can we get an advanced copy?
In writing the story, I found throughout my teaching experience, really, maybe you guys can give me feedback, really helps people to understand all the concepts and how to use them to explain how a real biological phenomenon works. So, we'll go through that. Maybe you guys can give me some more feedback on that later on how that's working for you. Wow, that's a lot left. Pardon me. Okay. Okay, but the first part of the story, I've said that part of the story like a billion times, right? That is a slam dunk for you guys, right? Okay, so I want you to tell me that. Talk to each other real quick. What's that first part of the story? How does that bug get in and start to grow? Not grow yet, but start to grow. Okay, get to the right environment. Talk to your neighbors real quick. I've said that a billion times. You guys probably have that already. You don't need me to tell you, I can say again. Oh yeah, you can introduce yourselves too. You may not have met everybody. So we have a mix of classes here today. It's kind of fun. All right. Oh, there you go, Brian. Okay, what's the first step? Attachment. Okay, how does the bug attach? Through binary fission? Now that's, what's binary fission? Hang on. Right, what is it? So it's colonization of the bugs. Right, right, so it's how they grow. It's binary means two, fission means to break apart. And so they're growing by this process called binary fission. That's how they multiply. Yeah. So eukaryotic cells grow through mitosis, and bacterial cells grow through binary fission. Okay. So we're trying to go through binary fission. Of what? Yeah, sure. Yeah, and I got a copy of here too. Oh, stage one. Oh, goodness gracious. Where's stage one? I don't know if I have a copy of that with me. It's week 2. What? Week 2. Week 2. That's where the cell structures are. Is that cell structures?
Yeah.
Wow. Excellent.
Yeah.
do what that looks. Good, good. Yeah? What's the normal microbiota doing? Yes, how? Uh-huh. That's right. Toxic products. Yes, that we should label them toxic products next time. We also have to get past the immune system. Absolutely. We're going to talk about that in our next unit. Okay, and what else? Yeah, what about the environment that the microbiota is doing? Yep, got to find a place. Find nutrients. Because you guys, the normal microbiota, you're using up all of those nutrients, right? Okay. Atmosphere. That was oxygen. Water. Okay. Wow, fantastic. You guys just got us to the point where we're happy, we're ready to start going through binary fission. Okay, great. Hey, Jen, we're going through the whole story, and I have said, you know, this first part a billion times, and so we're just having everybody else do it. To the starting of binary fission. Now that we've found a place at the table, and the environment is right, now we got to take nutrients in. Harvest the energy, make precursor metabolites, take those precursor metabolites, make macromolecules, make another cell. Got another cell. Okay? All right, great. So, let me go to these guys. Hi. Okay, so, guys. So, well, let's go here. Tell me what's the story.
Okay, so
There's a bug
There's a bug
out here.
out here.
Mhm.
And there's 3 ways you can adhere.
Okay?
You can adhere to the line,
Okay?
to cell membrane, or cell
So, wall.
Cell
Mhm.
wall. And there's everything else in the capsule.
Nice. Okay. Okay. So I'm the bad guy. I got my adhesins on my capsule, or my cell wall proteins, or my pili.
And you sat down there.
And there's 3 things that
Okay?
Let me contaminate myself.
Ah! Okay, Mara, sorry.
Competing microbiota, they can do.
That's you guys?
Yeah, they can send toxins.
Toxin.
Ah, toxins, okay.
The pathogen may or may not be able to attack. Because there might not, because they can take up the other bugs when you're taking up surface space.
Right.
And
I want to
then
multiply. I
they
could add
can
more
also
space.
be taking up nutrients, depending on how much they're.
Fantastic. All right, great. Okay. So now I found a seat at the table. Yeah, I'm still fending you guys off. What else do I need in order to be able to grow?
Temperature.
Temperature? Yeah.
pH balance,
pH,
the right
Water.
oxygen environment,
oxygen environment, temperature,
nutrient source.
and nutrients. Fantastic. All right, good. I didn't need to go through that. You guys got it. Fantastic. All right, so now this is the big one, right? It's now blab first, right? Blab first.
Mm-hmm.
Okay. So I'll blab first, and then you guys will be like, I already know that, and then you will work on your whiteboards and do it yourself. Okay? I'll do facultative anaerobe, and I am so going to, if I can, put that on the web for everybody. Sweet. And then you guys will do obligate aerobes and obligate anaerobes. Okay? And I'll get the chance to walk around and actually talk to you. This is great. Okay? So let's do that. We've got, here we go. Here is our cell wall. Here is our cytoplasmic membrane. Okay. And we are looking for how a facultative anaerobe is able to grow now that it's been able to adhere. It's found a seat at the table, right? It's able to fend off all the normal microbiota. It's got the right environment, the right pH, the right temperature, the right water availability. What am I missing? The right nutrients, and the right oxygen atmosphere. Okay? So our facultative anaerobe, you guys know already that it can grow with oxygen got it. Or without oxygen, okay? So let's assume that there's oxygen in our environment first, okay? Oxygen can transport across the cytoplasmic membrane by simple diffusion. There you go, simple diffusion. And in our cytoplasmic membrane, let's draw a couple of things here. This is our phospholipid bilayer. Here is our transport proteins. Okay, there's another one. Do more of them. There we go. Okay, so we've got 4 different transport proteins, all right? Dr. James, real quick, what kind of transport are we going to be able to see our cell using to get nutrients from the outside to the inside of the cell? Okay, what are the 4 ways that nutrients, which are too big to go through this phospholipid bilayer, they need a transport channel? What are the ways that our nutrients can go through? You guys know each other, right? Yes, yes. Okay, you guys working on it solo? Sweet. Yeah, yeah, cool. You got it?
The broad categories for examples. So the broad categories. I'm guessing if there's 4 types. Yeah. It is the phosphate group originally came from ATP, but it's a different ATP phosphorylated different protein, and that phosphorylated protein donates its phosphate to the phosphate. Okay, guys, what do we got? Yeah. Let's do that. Let's go through that. Hey guys, so we had a good question. So first of all, tell me what kind of transport. Well, never mind. Let me do that here. We've got simple, and then I'll let you guys fill in the specifics. We got simple diffusion, right? We've got Nope, that's not what I want. Hang on. Rewind. Types of transport. Transport that doesn't use added energy, transport that does use added energy. We're going to go back even more, okay? So, types of transport. We've got passive transport. And that is no required added, make sure it's added. It has its own energy in the system. We don't have to add energy for this to happen. Added energy, okay? And as a result, what is our concentration gradient for passive transport?
High to low.
High to low.
From high to low, right. Here I've got my perfume bottle, really high concentration here, and over time, I don't have to add any extra energy. The molecules are just going to, by Brownian motion, swirl out to low concentration. Okay, so it's going to go from a high concentration to a low concentration. Okay? Right, that's passive transport, the overall definition of passive transport. And now, yep, active transport. Let's look at active transport. Active transport requires energy for this to happen. Okay, so we have to require added energy. And the reason is because now we're going against that concentration gradient. Okay, now we're going from low back into high. Okay, so we're going against the concentration gradient. Okay, so again, using the perfume analogy, that's like me standing here and being like, okay, come on, all you molecules over there, get back in the bottle. What did I have to like vacuum them up or something, right? I have to use some energy in order to be able to get them back in the bottle, okay? So I have required added energy for active transport. Now, what are some modes of passive transport? You guys come up to what?
Isolated.
Facilitated diffusion. Facilitated diffusion.
Simple diffusion.
And simple diffusion. Did I spell that right? Okay, so we got one. That's simple diffusion and facilitated diffusion. What's the difference between simple and facilitated?
Simple is just small enough that it goes through the membrane.
So simple, this is our membrane. Simple diffusion, you just go right on through the membrane. Okay? Facilitated diffusion, on the other hand,
You gotta go through a channel like a protein.
yeah, it's too big to go through that phospholipid biolayer. It needs help. It needs to be facilitated. And so it requires a transport channel in order to go from high to low. Okay? Does that help, Farley?
Yes,
So simple diffusion, where it just goes right through that membrane, facilitated diffusion, that needs some help. It needs a transport protein to be able to go from high to low concentration. Okay. Now our bugs don't really use facilitated diffusion a lot. Because you remember that I'm the mean dude, and I've sat down at the table, but I've got competition for nutrients with all of you guys. Okay? There's not a whole heck of a lot of high concentration nutrients out there because you guys are soaking them all up. Okay? So I've got to find some other ways to be able to help me to grab the nutrients when I can. And so active transport is really usually the mode that bugs use in order to get their nutrients. Okay? So what are the modes of active transport?
with an ATP.
Okay, so we've got one that uses ATP energy, and that is our ABC transporter. Okay, what's the other one? Yeah, that's the one where we're using the proton motive force
What is proton?
in order to add that required energy. Okay, there's one more type of transport which uses energy, but is going from a high concentration to a low concentration. So it's kind of a mesh between the two. Okay? Yeah, that's group translocation. So we'll put group translocation in its own special category here. And what I'll do is I'll write it up over there on the board, a little graphic too. Okay? So then we've got group. Group translocation, which is using energy, but often going from high to low. Not always. Can go either way. Okay. So we come back over here. Yes, Elliot.
I wanted to, for the group translocation, this happened, like, toward the end of the proton force toward the end there, where you said that there's like a bunch of high electrons on top, and there's a spigot
Protons.
down.
Yeah, protons.
Up
Mm-hmm.
there, and then you had described it as like a spigot, turning it on.
Yeah.
Is that what this is? Is that what?
So group translocation uses ATP energy instead of proton motive force energy. And so let's go through that real quick, all those three different kinds. And sure, okay. And then what we'll do is we'll disband, and you guys can get out your whiteboards and practice it yourself. Don't look at the paper, and make sure you can do it on your own. If you can't, call me over, talk to your neighbors, okay? All right. So we've got 4 different transport channels here, okay? And we've got oxygen here, small enough to be able to go through, excuse me, our phospholipid bilayer. Okay? So oxygen, this is simple diffusion. Then we have, here's one of our nutrients, we'll just call them triangles, and it's going through by, well, there's a high concentration of triangles out here, okay? Low concentration of triangles here. So this would be facilitated diffusion. Okay?
And that's because it's using the channels.
Exactly. It's too big to go through that phospholipid bilayer and get through that hydrophobic barrier right here. And so it needs some help, even though it's still going from a high concentration to a low concentration. It needs a door. You know, we're too big to get into the room. You know, I can't get in. But if we have a door, we can open it up and come on in. Okay? So we also require facilitated methods in order to get in. Okay, so that's facilitated diffusion. Let's go jump to another color now. For the other 2. The other 3 rather. So these are our passive transport mechanisms. Let's go to our active transport mechanisms. So now we have nutrients that are at low concentration on the outside. We're trying to concentrate them inside because we want a lot of nutrients inside the cell. Okay, so we're trying to concentrate those nutrients inside. So now we have nutrients from a low concentration, trying to shove them into a cell where there's lots of high concentration of it. So we need to use energy in order to do that. So here, our circles are going through a transport protein. And we know now that there's 2 different places that they can get the energy to do that. Okay? We can use ATP. So here's our ATP. And as the molecule goes through, we're going to use that energy in ATP and break it down to ADP and inorganic phosphate. Okay? So we're going to use that energy to transport molecules from an area of low concentration, shove it into an area of high concentration. Okay? Does that make, yeah? Okay. We're going to use that energy that's stored in that bond between ADP and that 3rd phosphate group. Okay? Sorry?
This is what?
Ah, so this is active transport, and it is ATP-mediated active transport. Maybe what I'll do, I am getting 2.
ABC.
Yeah, it's the ABC transporter. It's the ABC transporter.
I'm getting a little too boxed in here. Move this one out here. Okay. Yes, so this is our ABC transporter. And it is an active transport. Using ATP energy to move molecules from an area of low concentration to an area of high concentration. Okay, what's our other mode of, move this over, of transportation, active transport, and that is moving molecules again from an area of low concentration to an area of high concentration. Now, we're going to utilize that huge concentration of protons on the outside. It's our huge reservoir, right? And so if we've got this huge reservoir here, we can turn the spigot, we can open up that channel and allow those protons to whoosh, rush through. As those protons are rushing through, look at the paddle wheel in there. I should get one of those things. Those little kid toys where you push one through and you see the paddle wheel turn. Get the paddle wheel turning, and as the paddle wheel's turning, it allows other molecules to come in with it. Okay? So we've expended some energy, we've released some of that potential energy that's stored in that proton reservoir, but in using that energy, we've transported some molecules in. Okay? It's that revolving door in the hotels. Okay? So this one, we are utilizing a huge store of protons out there. The protons are going from an area of high concentration to an area of low concentration. Okay? Through a channel. So the protons are using facilitated diffusion. But as we're using that, we're going to harness that energy that is expended through facilitated diffusion, putting that paddle wheel in so that we can grab, a red square, and push it through. Okay? Therefore, concentrating those red squares inside our body. That is the proton motive force.
It goes to high to low.
The protons go from high to low. The squares that we're transporting go from low to high.
So then just like facilitated diffusion, or actually the opposite.
The protons are just like facilitated diffusion.
But it's pushing protons.
But it's pushing protons. Yeah. Protons are using facilitated diffusion.
Sorry.
I can't say anything. There we go. We're harnessing the energy of that proton movement from an area of high concentration to low concentration and using it to transport material from an area of low concentration to an area of high concentration. Yes, Colleen?
So it's only using facilitated diffusion as it goes back into the cell.
For the protons. But since we're talking about transport of these squares, this is a method of active transport because we're harnessing the energy in that proton gradient in order to actively transport these squares. We need the energy here, so added energy is required, and we're transporting molecules from an area of low concentration to an area of high concentration. Okay? Yeah.
And you're,
No? Right.
the protons, they're going to do simple diffusion at the same time, right, through the membrane? But just-
Protons, even though they're really tiny, very, very strong positive charge,
so they
can't make it
they
through
go
the membrane.
right through the fire, okay.
So they have to go through a protein channel in order to get through. Yes, so protons require a facilitated diffusion or active transport. They require a protein transport channel. Okay, so Elliot, we've got both facilitated diffusion and active transport going on here. Okay. So in order to harness the energy from this proton gradient, we're going to open up the spigot and allow the protons to go from an area of high concentration to an area of low concentration. We don't have to add any energy to that. We're opening up the perfume bottle and we're letting them transport through, okay? But as we do that, we've got this huge reservoir. We open up the spigot, they're rushing through. We got a paddle wheel in there too. It's still the same protein.
So it's a channel for the protons to go through, and there's a paddle wheel in there. There's a revolving door. That revolving door is going to grab those red squares and push them through from an area of low concentration to an area of high concentration. Okay? And so we're using energy from this proton motive force in order to pump red squares from an area of low concentration to an area of high concentration.
How did it all of a sudden became a low concentration? Because I thought there was a bunch of protons above. Your spigot's open, going
Yes.
down.
Yes.
So isn't it high concentration above?
It's high concentration of protons, exactly, above.
Right.
And there, we open up the spigot, and they're going to run from an area of high concentration to an area of low concentration.
So that would be a facilitated diffusion type of.
For the protons only. Yes. Yes. We're going to use that energy. Yes, Mara?
The squares that are off.
So the squares are going in the opposite direction. They're going against their concentration gradient. They're going from an area of low concentration to an area of high concentration. Okay? And so we're going to use the energy as we've opened up the spigot for those protons to come through and use that revolving wheel to grab those rectangle red squares and push them through. Yeah? We'll keep working on it on our whiteboards? Yes. Okay. And so this is our proton motive force. Proton motive force. And is another mode of active transport. Okay. All right, we're almost done. Our last one, group translocation. Okay. Group translocation. Let's do a different color so we can follow everything. Can you guys, Brad, can you see okay over there? Kind of way over here.
That's all right, I can.
Okay.
I'm lower than that.
Try to put it up here. Except that I'm short. All right, so here we go. This one is where we're taking these triangles and circles and squares. Diamonds. I love it. All right, diamonds. Here we go. Now we're trying to transport diamonds into the cell. Excellent. All right, and this is, we have, get this here. We have another protein in here which has a phosphate group attached to it. Okay? And as those diamonds come through, that phosphate group is transferred to the diamond. Okay?
Is that inside the channel?
It's right here at the end of the channel. It happens right there at the end of the channel. And so as a result, let's see what is a better way to actually say it is to do this. So this square with the phosphate group gives the phosphate to the diamond, resulting in a square without any phosphate on it. And the diamond with a phosphate on it. Okay? So the phosphate has just gone from our square to our diamond. And now, as those diamonds come through, oops, that's not a diamond. There we go, that's better. We've got a lot of diamonds that have phosphate groups on them. Okay? That is group. Group translocation. Okay? All right, guys. Why don't you go ahead and get whiteboards out? Don't look at this. I wish I could put this behind somewhere. Don't look at it and see if you can do it yourself. Okay? All right. Go ahead and get them out.